Optogenetic Stimulation of Neural Subsets by Belgin Yalçın. Introduction. Optogenetics is a cutting-edge neuroscience tool which scientists use to excite or inhibit neuronal firing in awake live animals. By expressing light-sensitive ion channels, called opsins in neurons, we can control neuronal activity by light and using small optic fibers implanted in the brain. Controlling neuronal firing allows us to study the relationship between activities of specific neurons and behavior. This technique extends across a variety of model organisms, including fruit flies, zebrafish, rodents, and non-human primates, for investigating neuronal activity and brain physiology. A broad range of research areas, from systems neuroscience to neurological and psychiatric disorders, involve applications of optogenetics for the recent seminal discoveries. Opsins are ion channels that open or close in response to light at specific wavelengths. They are naturally found in microbes but not in vertebrates. Depending on which ion an opsin channel gates, its activation by light can either depolarize or hyperpolarize a cell, thus excite or inhibit a cell. For example, channelrhodopsin is an excitatory opsin, while halorhodopsin is an inhibitory opsin. Researchers continually improve and expand the opsin toolbox by engineering naturally occurring microbial opsins and modifying the existing ones. Channelrhodopsin is one of the most commonly used excitatory opsins for optogenetics. Blue light at 473 nanometers prompts conformational changes in channelrhodopsin and allows diffusion of cations into the cell, which in turn causes rapid depolarization of neurons that can lead to action potentials. Optic fibers implanted into the brain allows researchers to stimulate opsins like channelrhodopsin with light while an animal is awake and performing a behavioral task. We can control which neurons are stimulated by where the optical fibers are placed in the brain, and also by using genetic tools to drive expression of opsins in specific subclass of neurons. Next, we will see an example for an optogenetics experiment. Methods. Design of an optogenetics experiment involves a few steps and considerations. First, what are the neurons of interest and how can the expression of an opsin be targeted to those neurons? Second, where the optical cannula should be placed. This component can also add another layer of specificity. Third, how should the stimulation be performed and what behavior is expected as the output during stimulation? Here, we will activate neurons with channelrhodopsin and blue light in the mouse premotor cortex. 1. Expression of opsins. Virus-mediated gene delivery is one of the most common ways of expressing opsins in the brain. We inject an adenovirus expressing channelrhodopsin from an HSIN promoter into the mouse premotor cortex. Here, you can see an example of this procedure. We express channelrhodopsin in the mature neurons in this brain region and target these neurons with light and activate neuronal firing. Most opsins also carry a fluorescent marker, such as yellow fluorescent protein or YFP, so that they can be easily visualized under a microscope after the optogenetic stimulations to confirm that channelrhodopsin expression is successful. In this microscope image, you can see a successful channelrhodopsin expression in the premotor region in green. Also, YFP expression alone without a channelrhodopsin should be used as a control for comparison in these experiments. Additionally, there are other methods of opsin expression, including transgenic mice lines. In these animals, opsin expression is permanently integrated into the genome with the use of cell-specific promoters to focus their expression in specific neurons. 2. Optic fiber placement. Placement of an optic fiber facilitates light delivery from a light source into specific brain regions. When the fiber is attached to a laser light source, we can send signals to the tip of an optic fiber. Thus, channelrhodopsin expressing neurons at the optic fiber tip receive the light signals and depolarize. Fiber placement is performed with stereotactic intracranial surgery in compliance with animal protocols, and the implant is secured to the skull with dental cement. These examples show placing an optic fiber into the motor cortex, and you can see the mouse in recovery after the procedure. Light delivery into this region allows us to excite the channelrhodopsin expressing neurons specifically. 3. Optogenetic stimulations. For successful activation of opsins, we need to use a light source at the appropriate wavelength for gating the ions with high efficiency. Here we're using a blue laser light source at 473 nanometers wavelength to stimulate neurons which express channelrhodopsin in the premotor cortex. You can see this mouse starting to move when the light comes on. Activation of premotor cortex neurons in one side of the brain elicits this movement towards the opposite direction, indicating a successful optogenetic stimulation. In comparison, control animals do not display this behavior as their neurons only express YFP, not the ion channel channelrhodopsin. Conclusion. Optogenetics allow precise control of neuronal activity in awake behaving animals. It's a powerful technique to dissect out the functions of neurons and the relationship between neuronal activity and complex behaviors. Ever-expanding optogenetics tools, together with a diverse array of neuroscience methods, immensely widen the horizon for investigation of brain function and disease.